This video will provide an overview of arguably the most extreme and final cell signaling pathway that exists, apoptosis, a form of programmed cell death. Programmed cell death is caused by the cell's own intracellular signaling components, as opposed to death caused by injury or other insult from the outside world, which causes necrotic cell death. Programmed cell death occurs in a wide variety of organisms, including bacteria and plants. Apoptosis is a version of this cellular suicide that's specific to animals. Just as an aside, pronunciation of this word varies from apoptosis to apoptosis to apoptosis to apoptosis. All of these are commonly used, so just say it the way you like. Necrotic cell death can cause damage to surrounding cells. For example, if you burn yourself, the cells that die as a result break open, releasing potentially damaging enzymes and other substances which can then harm nearby cells, causing more damage. In contrast, apoptosis is a clean death, in which the cell essentially tears itself apart on the inside and fragments into vesicles, but the contents of the cell are contained within the vesicle blebs. You can see what this looks like in this image on the right. At the top is a normal cell, and at the bottom the cell after apoptosis has destroyed and fragmented the cell into membrane-enclosed compartments. So what's the role of apoptosis? Why would a cell ever voluntarily remove itself from the organism? Turns out this is a normal process that happens both during development and in adulthood. It's happening in your body right now, in fact. Each of us loses upwards of 50 billion cells a day to apoptosis. It's one of the main ways our body gets rid of worn-out cells and makes room for new ones. During development, when tissues are refined and remodeled to form functioning organs, part of that refinement process is getting rid of unneeded cells. An example is shown here at the bottom with digit development during embryogenesis. Initially, we start with a limb bud, a lump of tissue that will eventually be the paw. This is a mouse limb bud, but the process is the same for our hands. To form individual fingers, the cells between the digits are stimulated to undergo apoptosis, leaving just the digits. We also see hormonally induced apoptosis occurring in the uterine lining in females and in follicles in the ovary throughout the reproductive lifespan. Apoptosis is also responsible for tail regression in everything from human embryos to tadpoles. Apoptosis can also be triggered when a cell has fulfilled its purpose. For example, during the inflammatory response, large numbers of immune cells migrate to the site of inflammation to fight off bacteria and other pathogens. As the inflammatory process winds down, these cells undergo apoptosis. Some types of immune cells can also trigger other cells to undergo apoptosis if these cells are displaying abnormal molecules on their surface, which happens in the case of viral infection, as well as with cancer cells and transplanted cells. The immune cells detect that there's something weird about the cell, and rather than risk potential harm, stimulate the cell to remove itself from the population. Cells are also capable of initiating apoptosis on their own, if their own internal sensing mechanisms detect that particular types of cellular damage are accumulating and becoming potentially problematic. This includes DNA damage that can't be repaired or resolved, extensive protein misfolding within the endoplasmic reticulum, or accumulating as aggregates in the cytoplasm, and shortening of the chromosome ends, which are called telomeres. A cell with a functioning apoptotic pathway will destroy itself, preventing harm to the larger organism. Obviously, mutations that disrupt the normal apoptotic signaling pathways can cause abnormal cells to persist in the body, and this is one mechanism that can lead to abnormal cell division and eventually cancer. But assuming that the apoptotic signaling pathway is functional, how does it work? How does the cell tear itself apart? Whether the signal that initiates apoptosis is from the outside world, say a death ligand on an immune cell is telling a virus-infected cell to delete itself, or from damage control signaling pathways within the cell itself in response to something like shortened telomeres, the molecular mechanisms are similar. Regardless of what initiates it, apoptosis is a one-way process. Once it starts, it can't be stopped, so it's very highly regulated and controlled. Apoptosis often involves damage to mitochondrial membranes, causing them to release more apoptosis-promoting molecules into the cytoplasm. Typically, apoptosis also involves activation of intracellular proteases called caspases. Proteases are enzymes that break apart proteins by hydrolyzing the peptide bonds between amino acids. There's a whole family of caspases that get activated during apoptosis, and these in turn activate nucleases that break the phosphodiester bonds in DNA, resulting in fragmentation of DNA in the nucleus. The caspases also directly degrade cellular proteins, like those that make up the nuclear lamina, which we've learned is critical for maintaining the shape and structure of the nucleus. This results in the nucleus breaking apart into smaller compartments. Caspases also degrade cytoskeletal structures like actin microfilaments, which are critical for maintaining the overall shape and structure of the cell. And when this happens, the cell itself begins to fragment. This massive change in cellular structure releases molecules that can attract phagocytes to the tissue containing the apoptotic cell, so-called find me signals. And the fragmented cells display abnormal lipids and proteins on their membranes that function as eat me signals. Once the phagocyte encounters these, endocytosis of the apoptotic cell will occur, and the cell fragments are thereby removed from the body without damaging any surrounding cells or tissue. This electron micrograph shows a neutrophil, which is one type of phagocyte in our immune system, indicated by the black arrow, engulfing an apoptotic cell, indicated by the light arrow. These and other types of phagocytes are constantly surveilling tissues and getting rid of this type of cellular debris, serving as the body's cleanup crew. I've given you a link to a beautiful animation created by Drew Berry that shows the typical molecular events that occur in a cell undergoing apoptosis in response to a death ligand from a T cell. It's in the supplemental videos page on our Canvas site. I'd encourage you to watch it. As you watch, note how this is also an example of signal amplification, which we've seen as a general feature of signal transduction pathways. As I mentioned, cancer cells often have mutations that disrupt normal apoptotic signaling, making them resistant to apoptosis. One gene frequently mutated is called BCL2, which encodes a protein found in the outer mitochondrial membrane. The mutation causes too much of this protein to be made, so that it impedes apoptosis. A recently developed drug called venetoclax targets this specific protein and is showing promise as a potential chemotherapy for these cancers. Here's a link to another animation, this one made by Etsuko Uno, showing the molecular mechanism by which venetoclax targets the BCL2 protein. It's also linked on Canvas.